Well, hi, my name is Mallika Nocco, and I am an assistant professor of cooperative extension in soil plant water relations and irrigation management out of UC Davis. And I'd like to introduce Logan Ebert, who's not yet mic'd, but coming to the camera too. Logan Ebert just passed his qualifying exam. So he's a dissertator in our research group, and we're really proud of him, and he's doing a lot of fantastic work in drones. So together, this is our first year doing this in person, so we're excited to see all of you, and still excited about those of you who are on camera. But it's just kind of nice to also be in person. And we're going to talk about thermal imaging and crop water stress, which is what our lab really focuses a lot on. And are the slides all G to G? Oh, okay. Okay, we'll just hang out. That's cool. Who has a thermal camera? Does anybody have a thermal camera? Whoa, okay, so a lot of people have thermal cameras. I mean, I would say that was what, 15%, 20%? Okay, that's really exciting. Has anyone tried to map evapotranspiration or crop water stress already? Mark. Yeah, so too, can you share some of the approaches you're using? Okay, so that makes sense. Okay, cool. One last thing before I jump into the slides. I know that everybody fills out surveys about what we're doing. We've been toying with the idea of doing like a DroneCamp adjacent or DroneCamp related longer crop water stress workshop, because this is 50 minutes. So we're going to talk a lot about theory and ideas and applications that we're doing, but we're not having you, work with data and map anything and actually get into it, right? I want to know if you guys are interested in doing that. Oh, okay. Well, if you could also write it on the survey and tell me whether you would be up for a half a day or a full day, and whether you would prefer remote or in person. That'd be helpful for me just in my long range planning of developing something like that, because we've been talking about doing it. And it was nice to just see right here that there's a need. But anything else that you would like to tell me about how you would like that to look in terms of delivery, amount of time, etc., that would be good. And one last question for you, who, in terms of coding, because you can't map crop water stress or evapotranspiration or any of this without getting a little bit into coding. How many folks are familiar with R? Hey, that's good, that's good. How about Python? Okay, it seems like the same hands went up. Does anyone have a preference for R versus Python? Like, how many people would prefer R? Okay, how many people would prefer Python? Okay, so really no information. Awesome. All right, well, thank you for that mini interaction, and we'll get going. Okay, so we're going to be talking about sensing crop water stress with thermal imagery. And here's just kind of a roadmap. So first, we have to talk a little bit about evapotranspiration, or ET, and as well as crop water stress. We're going to go into some of the limits and applications of UAVs or drones for mapping crop water stress or crop water use. And then just talk a little bit about the useful types of imagery, as well as some different models, and then go into case studies with the models that we're using right now in our research group. And then just having a conversation about where we're headed. And if anyone wants to share challenges they're having or ideas about how they want to use it, I'm hopeful that we'll have some time to actually do that. So a little bit more about our research group. So we call our lab the Conservation Irrigation Lab. And there's really 3 areas of work that we're focusing on. Optimizing irrigation for co-benefits. You can see there's deficit that's crossed out right there. My job title was actually deficit irrigation. But farmers do not like to hear the word deficit because they feel, is anyone a farmer in here? Oh, okay, I was gonna pick on you a lot. Oh, well, they, in a good way, to just, bring this up. They feel that you're telling them to cut yields in order to conserve water right away. So for me as an extension scientist, it was like going into the room punching without even knowing it by having that word up. So I've been switching and thinking more about optimizing irrigation. Because really, for growers who are thinking about this, it's about optimizing yield, optimizing water usage, and potentially optimizing quality of fruits and vegetables and other crops. We also are thinking a lot about regenerative irrigation, which is thinking about how you can maintain and promote soil health and soil quality and carbon sequestration and microbial communities and agroecology, while at the same time conserving water. So that's another area that our lab work focuses on. But almost everything you're hearing about today from us is this idea of precision management and monitoring, because that's where the drone mapping becomes particularly exciting in agriculture, right? Is when we think about ways that we can maximize and optimize the application of inputs, whether that input is water, which is what we study, or nitrogen, or pesticides, or other types of inputs that you're concerned about. So, what happened there? Oh, here we go. So evapotranspiration. I really like this cartoon, even though it's like for children, because it's so intuitive. So evapotranspiration is evaporation and transpiration. So you have evaporation from leaf surfaces and from stems and from the soil, and transpiration is water moving out of these little pores called stomata on leaves. And that transpiration, when you water a plant, 90% typically of the water, this is any plant that you're putting on it, is going to this transpiration process. And that's because when a plant wants to get carbon dioxide, it has to open up these stomata, and carbon dioxide diffuses in. And the cost of getting that food is a loss of water. And that's just kind of how this goes. So that's ultimately, that's crop water use. So when people talk about crop water use, they're oftentimes thinking about evapotranspiration, or ET, and that's the acronym that we use for it. And I just think this is neat. It's like, try this at home. If you cover it, you'll be able to experience ET. So that works. I've done that before I put this slide up. And thinking about ET at the landscape scale, right? Like, it can really have a powerful impact on a mapped landscape at whatever scale you're looking at, whether it's precision with a drone or through satellites. ET can change the water cycle when you have these expanses of continuous irrigated landscapes, which is another thing that I'm super interested in. There's a lot of ways to measure ET and actually get at it. And there's lots of different flavors of ET. And I just want to bring that up because it can come up when you start trying to map this stuff. And if you're just doing a lot of like self-driven research and looking stuff up, you just want to know what you're looking up and like what algorithm you're going to maybe try, right, with your imagery. So potential ET is this theoretical maximum. So what is the evaporative demand of the environment? And they use all these pan evaporation methods to try to get at them. And there's also a lot of empirical equations that are used to understand potential ET. Reference ET, how many people in the room are from California or doing stuff? Okay, who's all heard of CIMMYS? A couple, okay. So CIMMYS is, everyone should know about CIMMYS. It is the California Irrigation Management Information System. And that sounds like, okay, well, I'm not interested in irrigation management. Why do you need to know about CIMMYS? It is a really broad network of weather stations throughout the state that's maintained by the Department of Water Resources. And there's what I love about this. And if you're from another state, like I came here from the Midwest, and man, I would have killed for something like CIMMYS, just because there's such a standard in terms of how these MET stations are all maintained. They all have the same sensors, and they're all really developed and created so they can measure reference evapotranspiration. And reference evapotranspiration is another like index of the evaporative demand of the environment, but it's one that's kind of amassed a lot of like research capital over the last few decades. And we have this whole kind of algorithm system as irrigators for thinking about how much water a crop is using and thinking about the crop water requirement. And that all uses this index, this reference ET, or sometimes you'll see it with the ET and then this is 0 here as a subscript. And so it's hypothetical ET, and it's calculated over a short grass, well-watered surface. And basically what it does is, using this empirical equation takes away a lot of like the varying plant physiology of crop water use. So we use that pretty often. And a lot of the drone mapping algorithms are somehow kind of connected to reference ET. And then there's actual ET, which is tough to measure, right? So you can measure the actual amount of water that's being lost from a crop canopy. You can do it through this, through lysimetry. There's not supposed to be a T right there, which is basically a giant weighing device that is put into the soil, and it's like married to the soil, and it changes its weight oftentimes, depending on how much water is being lost from that soil monolith. You can also measure ET using these lysimetry devices in more humid areas by doing a water budget and measuring the drainage that's lost. You could actually do that here too. There's just not a lot of drainage. But one of the other ways that maybe people will see in California, there are quite a few of these eddy covariance flux towers, and they use energy balance methods and models to estimate evapotranspiration or crop water use. And those are wonderful, and it's one of the best tools that we have, but they have they require a large footprint. And it's like almost, you could think of it as about the scale of a field or a little bit less than a field, like a quadrant. It depends on how tall the tower is, what that footprint is going to be, but it's a uniform measurement that's continuous in time. So you'll see that oftentimes, here's a photo of a flux tower. This is a pretty short one because it was over tomatoes or potatoes. And that we couple them with drone images, because one of them is giving us this really accurate time series estimate of crop water use. And through our drones, we're able to get a really wonderful understanding of the spatial variability of crop water use. And if you're working with drones, you're interested in some sort of spatial variability of something. All right. So, and then this is just the crop water coefficient method if you want to estimate actual evapotranspiration, which is something that many folks are using here in California. So this is just kind of how I think, and we as a research group are thinking of all of these different ways of measuring and managing crop water and just the needs of the state, right? So we're thinking at this subfield scale when we're talking about drones, and that's all precision management and monitoring. But it's really important if you're going to get into the crop water use game, or just water, especially water in the West, to think about all of these other scales, right? So there's the field and the farm scale, and they oftentimes have goals thinking about controlled deficit irrigation, water use over the course of a season, many different crop rotations. Individual growers are dealing with the drought. They're dealing with different constraints on water availability that are seasonal. And then there's this policy interest where we're talking about, managing recharge, thinking about groundwater banking, managing water use over the coming decades, Sigma. All of this stuff is active and happening at that larger spatial scale and at that decadal and multi-decadal time scale. And there's different tools, right, for all these scales. And I think UAVs is a great tool for this subfield scale, but they can still kind of play with this other stuff, right? Like, I think we still need to think of all of these things together and collaborate with different groups of people. And that's what we're trying to do. So what are the drivers of evapotranspiration? Well, the drivers of, I think we can come up with these, right? Like, what do you guys think drives crop water use? Sunlight, temperature, wind, and what we call the vapor pressure deficit, which is an understanding of the aridity of the environment. So these are all big drivers of crop water use. And usually in the algorithms that we use to map crop water use or to map crop water stress, these are often fixed, and they're not very, we are not measuring how these vary in space on a field. But we are getting information from a MET station that's close by. So precision irrigation and precision agriculture is becoming more and more technologically feasible. It's feasible for folks on the screen who are working in areas where there are these center pivot systems, where there is this zone-based control. Most of the irrigation in California is happening with blocks, right? So how small could a block get? Is what we're limited by. But now there's more and more tools like these variable frequency drivers, things that are making it more accessible and more of a reality that we can control water at smaller and smaller spatial scales. So the goal of what we're doing is to try to make sure that our information is in pace with the technological innovations that are happening to deliver the water at higher and higher resolutions. Okay, so once again, we got to get that scientifically based decision support in the actionable time frame and be developing our evapotranspiration, our crop water stress algorithms to be right at the level of high resolution information that is most available to us from our technology. So how are we going to do that with drones? And this is where I'm going to turn it over to Logan. Yeah. And you need to steal the microphone. Okay.
Be too low. I'm shifting. All right, can we hear me still okay? Awesome. All right, hello everybody. Yeah, now transitioning on to like why we want to use remote imagery. So like Mallika brought up earlier, all the plants have these stomata and they respond differently to this different amount of water stress. And that's what we're looking for is water stress in this. So when you have plenty of water, the stomata are going to remain open. They're going to be able to stay more open. They're stayed open by turgor pressure in the cell because they have all that water. But as they become more water stressed, it's not having the water to keep those cells open. So they start to close, they start to transpire less, and they're also getting less carbon as well. And we can use this remote imagery or thermal imagery to detect this, because when the stomatas are closed, you're not getting as much transpiration. Transpiration cools the surface. So you're going to start seeing an increase in temperature as you get more water stressed. I think we do this. And then going on to also, we use multiband imagery as well. So besides thermal, so the 5 and 7 range here, what is highlighted is where the soils go, or sorry, the thermal bands go. But on the left edge, that's where our multispectral imagery are. And that's our colors for the 1, 2, and 3 for our red, green, and blue. And then 4, we have our near infrared. And we can see, and these are just some vague approximations of soil and vegetation, but we can see some key differences in these ranges between these. And in particular, with the red, green, blue, and I believe as we go into this next section, when we look at vegetation, we see a difference between healthy leaves and unhealthy leaves. When you have a healthy leaf, you have this nice green color. And you can see that because you're having the highest reflectance of that green color there. So that's what's being bounced off from the leaf. That's why we're seeing that kind of green color. But as it becomes less healthy, we're seeing less of these values. So it starts to change color. But we can also see that the ratio between these different values are changing over time, or not over time, but as it becomes less healthy. And even to the end, when you get the dead leaf, and they're all kind of just smooth overall because it's just a kind of black brownish surface color at that point. So how do we use these bands though? Because just looking at, if we were to look between a healthy leaf and a dead leaf and just look at the blue band, it's going to be hard to tell the difference between the two. So what we use is something called vegetation indices, and there's a lot of them here. This is just to show that there's a plethora of different indices out there, and they all relate to some different part of the plant and what kind of reflectances you have off of that. But what it does is allows you to compare the different bands to each other and create some sort of ratio or relationship or an indice that will tell you some characteristic of its condition. And I'll be getting into a few different versions of these. One of the most common ones is the normalized difference vegetation index, or NDVI, widely used partly because, oh yeah, so that's good. Popular because it's fairly simple to calculate or to collect. It's just red and near infrared bands. It's used to look at the differences between that really highly stressed versus healthy plants. I guess if I were to go back quick to point out, you see this really high reflectance in near infrared when it is healthy, and then on it does not. And near infrared's not great for the leaf, so it wants to try reflecting that as much as possible, because that's what's doing damage to some of the cells. So as we go back to NDVI, as that ratio gets closer to the red, you get a lower value, and that's kind of indicating that you're having a more stressed plant. So NDVI goes from scale, when you're talking about vegetation, from 0 to 1 typically. When you're looking at more urban features, it can get into negative values. But typically, as you have a higher NDVI, you have a healthier canopy. One issue with NDVI, though, it is prone to becoming saturated, especially in high density canopies. So as you get many layers of leaves over each other, it ends up just absorbing too much of the light, and you're not getting great reflectance value. So as you say, you get into like potatoes or tomatoes later on in the season, when you have those really dense canopies, it can be not as reliable down that line. Which is where we get into this next one, Enhanced Vegetation Index, or EVI. Does anybody use EVI? Just to some people. I like EVI because it factors in a blue band, and this helps kind of alleviate that saturation. There's a bunch of different coefficients in there that are derived from different biometeorological equations. But it's the same principle of the lower values are more stress, the higher values are less stress. And I guess I should take a moment to explain some of the imagery here. This is over some potato data from Wisconsin. And you can see this middle area right here is a field road. This is a bare soil area. So you can see these are more stressed because there's no plants there, or even the ones that are on the edges here are becoming more stressed because they're right next to the road. Yeah, the center pivot track going around here. So this is where everything's getting stomped on by the center pivot. So not happy plants there. One last one, and this goes away from the multispectral imagery into the thermal imagery, is the crop water stress index. Has anyone used this before? So,
oh, okay. I'll just.
oh, it'll come over to the camera probably.
Okay. Can you tell us about your experience with the crop water stress index? Like, what crop did you use it on, and was it easy to use? Oh,
yeah.
Okay. What was your name? Oh my gosh, hi Brandon. Oh. Okay. So Brandon is an amazing graduate student at UC Davis, is using masks are so tricky. Is using the crop water stress index in almonds. And he just brought up that it's tricky because there are a lot of different terms. It looks innocent. It looks like a very innocent equation, but if you get into each of these variables, it turns into a whole, thing. I don't know, Mark, do you want to share your experience?
Sure. Well, I'll pass the mic around.
Oh,
Pass the mic. Okay. All right. No, just kidding.
The crops that I work with are actually often natural vegetation or restoration sites. So we're talking oaks, for example. So vegetation with really complex canopies cause a lot of different temperature variations that don't necessarily have something to do with ET or that, illumination and how basically the sensor geometry makes it complex to use some of these things sometimes.
Yeah, I don't know. Vegetation that is not one crop and a totally different canopy, I think that would be really, really hard to use with this in its current form and iteration for sure. But that's an interesting. There's potential. Yeah, that's super interesting. All right, back to you, Logan.
Simply what it is, it's looking at the average pixel or the temperature per pixel, and then comparing it to a wet pixel, which is a pixel that has or a theoretical wet pixel. Sometimes you're not going to have these images, especially or that data, because sometimes the fields aren't going to be able to be completely or get all the water they want. Especially if you're out in California, you're not going to have this idealized wet pixel. And what this is the lowest temperature at which this canopy is going to be. This is going to be the one that receives all the water. And going back to what I was talking about, how transpiration is changing the temperature. And you're comparing that to the dry pixel, where it's the highest temperature that a pixel or up the canopy will be, and if it is severely water stressed. And typically, a farmer doesn't want the plants to be at that level either. So there's a range of doing this empirically, like on the left, and theoretically on the right, do my sides right. Both these methods either try using things we find or using other biometeorological equations to solve for what those would be. What's nice about this, though, is this is just using the temperature band, and cameras have improved a lot. I'm sure you guys have learned a lot about different equipment here, but sometimes it can be difficult to get a mix of both cameras, and it all depends on what's in your range. What's nice is this is a single band you're trying to collect then, or a single camera. So a quick interlude for a joke here. What remote sensing allows us to do is tell what is photosynthesis and photosynthesisn't, if I can pronounce anything right. So something that's nice, though, when we combine both our multispectral and our thermal imagery, is we can solve or model evapotranspiration using a multitude of different models. So here's just a list of some of the more popular ones. I have no loyalty to any of these in particular, but just to say, there's metric, TSEB, or the two-source energy balance, and then CVOL. Does anyone use any of these for mapping ET? Who maps ET out there? Makes sense.
That's why we got
it. Yeah. Is anyone interested in learning how to map ET? Yeah. Okay. Okay. Yeah. So I guess, yeah, I'll go into some case studies of what we do with all these. Starting off with let's go over the equipment that we use in our lab. So this is our drone, Matrice 210, and from DJI, and MicaSense Altum camera. Does anyone use that? Is anyone? Okay. The Altum is a combined multispectral and thermal camera. I believe it was designed for agricultural purposes, and it's great, pretty straightforward of how to use, and it gives us great imagery. I feel like personally, but when we process our imagery, we use a workflow, and it's always good to have some kind of printout like this just to help yourself go along as you start to do this more and more. But basically, we take in the top left, we're taking all of our multispectral bands, and we have to mosaic them using some sort of program, whatever software you use for that in that particular case. For our thermal bands, we have to calibrate it from whatever the drone is measuring it in to a more usable measurement like degrees Celsius is what we use. The multispectral, we take and calculate different phenological aspects like the leaf area index, or how dense that canopy is, and canopy height. And then thermal, we just put in, and we'll take that all together, and that's what goes into our evapotranspiration estimates. There's some also meteorological data that we're bringing in the center there, and then there's also some literary values that we need, like soil emissivity and albedo for the soil and the vegetation. And they're constants we're using. We're not actually solving for those. Just going in a little bit of the model I use, the high resolution mapping of evapotranspiration, or Hermit model. I won't linger too long, but it's a two-source energy balance that solves for evapotranspiration as the residual of the energy balance. And what's unique about it is that it uses a iterative solution to solve for sensible heat flux, which is anyone who's all familiar with, I guess, the energy balance in general? And okay, good. Yeah, and this is something that in this workshop would be good to go into more, and how these are all based in, or a lot of these ET models are based in. But basically what's important about the iterative solution is it allows us to avoid that hot and cold pixel issue that we had with the crop water stress index, because it runs a lot of simulations kind of narrowing in on a point, so it avoids that. And the three main inputs for it are that met data, canopy surface temperature, and EVI. Is it Noah, or it's a
It's no, for our met data. I mean, the beauty of it is it can be any met data source, right? So we're just trying to get met data that has the variables that we need, that's as close as possible to the field, right? If you set up your own met station and it's right on side of the field that you're working at for a research purpose, that's really useful. One of the things I really like about the California Irrigation Management Information System is that if we are using and working to calibrate our models with the tool that's accessible and available to everybody, then we have an understanding for how the model is going to work in a way that everyone would be able to potentially use it. So we are trying to use the CIMIS stations as much as we can.
The next station is more than 25 kilometers away from where you're doing the work.
Yeah.
The data may not be
right. And okay, so the question was like, is the CIMIS data, which is our California Irrigation Management Met Station data, preferable if the stations are far, greater than 25 kilometers away? And I would say no. Like, and we've had to make that call before too. Like, we want to use them as best as we can, but when you start to get really far away, the met data is really important for getting back to what we talked about earlier, thinking about the evaporative demand, right? So if you get too far away, then that's not going to be indicative of what those crops are experiencing. So you have to convince yourself that the met data you're using is close enough where you're actually trying to model the evaporative demand. And that's the case for any algorithm that you're using. You probably are going to need these met data. There's none of these evapotranspiration mapping from a drone situations where you don't need to know sunlight or wind speed. So you're always kind of going to some sort of data source for that. But great question.
Yeah, and this particular one is not a CIMIS station. This is a public station in Wisconsin. As I go into what I study, and one of my papers that just came out this March is a study I did in potatoes and asking the question of how high to fly. So you can fly at a range of different heights with the drone, and I was trying to figure out what height would be best, because you can fly lower and you get much higher resolution, but then you have longer processing times and you have higher data storage, higher battery usage. So it's like, what is that balance of what you want? And do we gain anything from having that higher resolution imagery? So these are maps of evapotranspiration across actually, no, this is a map of relative evapotranspiration. It's ET normalized across the field. And we do that to really accentuate differences in ET. So we get really bright colors that we can see where different parts are. And going across is just different dates. And then from the top down is 90, 60, and 30 meters above ground level for our drone height. And what we ended up seeing is there wasn't a ton of difference. Like, you're not seeing a bunch of differences between the different heights on a given day. And what that tells me is, in this particular case, there wasn't a benefit to fly higher. Obviously, this is very dependent on what you're looking for, but this is a center pivot. And I guess in general, you don't need to fly lower than you need to get the finest resolution. So you don't need to fly really low to the ground if you're trying to get meter. If you can only irrigate to a meter size, you don't need 10 centimeter resolution because you can't irrigate to that. But continuing on with just more of the study here, we did some other fun analyses with inputs and some uncertainty. But that's can be more if you want to look into it. Ebert et al. 2022. Humble advertisement for myself, but I'll give it back to Mallika.
Ooh, okay. Can you hold that question? I have a couple of slides about that at the end that I want to show you. But great question. And we will get to that. Good. I'm back. All right. So this is just a great example of just some of the questions that we're asking, right? Like, how high to fly is really important. And one of the other kind of key questions of interest is, this was fine, and 90 meters was great for potatoes, but you know, for almonds, where you actually have a really high value crop and they're individual trees, and you might be interested in what a particular tree is up to, there may be more value in actually getting that high resolution down to that tree level. And we also have some work where we're trying to get to that tree level insight. So in this part, yeah, I just wanted to kind of share, this is really to just give you folks some ideas of like what you could do with thermal imagery, what you can do thinking about crop water stress and evapotranspiration. So this is a problem that we've been working on in tomatoes, and it's a response to deficit irrigation. And the data that I'm showing here are not from a drone. They're from a low-flying series imaging like plane, like a small Cessna that we worked with. This is data from the pandemic year when nobody could do anything, 2020. But I'm showing you the data to just kind of show you an example here of what could be done with a drone. And Logan's actually doing this with a drone this year. And it's neat because until we had the power to collect really high resolution thermal imaging, people would do studies, right? And you do a study and you do deficit irrigation work, and you take ground measurements, right? You go out, you take plant physiology measurements to see how are the crops responding to lower watering? What are we seeing in terms of yield at the end of a season? But with a drone or with this type of imagery, we can see when like before we started the deficit, no stripes, and then the stripes come. And that's super exciting. I got so excited for my sad little home during the pandemic year when I saw those stripes, because it showed me that there was a plant physiological response to our change in irrigation regime. So in a way, it's really changed the way that we can experiment with water use, having this additional tool to actually monitor the crop stress response. And that was super exciting. And this study is pretty neat because we're also looking at how differences in deficit irrigation impact human nutrition. So lycopene, which is an antioxidant that's found in tomatoes, vitamin C, beta carotene, really fun study. Another thing that we're doing that's been interesting is looking and working really closely with Professor Beth Forrestal and Andrew McElroan, who are in the viticulture and enology department and USDA, is how to irrigate during a heat wave. And this is similar kind of study in terms of the different irrigation blocks. So these are cab grapes, working with also in partnership with Gallo out in the Central Valley. And growers have about 10 days when they know a heat wave is coming. So what do you do in 10 days is the story of this study. And they're different blocks and they have different treatments. What often is done in that 10 days when you know that a heat wave is coming is let's put a ton of water down, like let's put all of the water on it, and maybe these grapes will be okay. But that wine did not taste good, the all of the water wine. And one of the things that we've been able to do with drones in this study is to try to, once again, understand what the response is physiologically. Where is the stress seen and where is it not seen once those different irrigation treatments are put down. So poor Andy, who's in our research group, actually had to go plan his missions for the heat waves in the Central Valley and go and collect imagery during the worst days. But he's taken heat management training, so he takes good care of himself. But this is kind of neat. It was really hard to interpret the first maps. I wanted to see, just like I saw those stripes, I wanted to see those little blocks, right? Like I wanted to see these blocks in the drone imagery, and we didn't see it. And we were just like, oh man, why didn't we see them? And it's because of the grapes. You know, the actual grapevines, when you see them from the imagery, they're not that much of the image. So what we actually had to do was some interesting thresholding work. And we used the NDVI and EVI of the imagery to look at where are the peaks that are indicative of the grapevines and what is like the low-lying cover crop or bare soil. And when we did that, then we could see the differences. So basically we had to cut out the interspacing, right? And then we could see the differences in the vine responses to the irrigation. So that's another cool study that's happening. Another way that we're using drones, and this imagery is actually from RGB imagery from a different drone, but still useful, and we're still flying thermal here, which is we are trying to understand how long you can wait before you turn on the irrigation on an almond orchard in the spring. Because many folks just do it when their grandparents did it and nothing has changed. But there's water that can be saved, and actually healthier almonds, because you want to make sure that the almond roots have enough oxygen to expand. And there are measurements that can be taken, particularly stem water potential, to try and aid in this information. But that is a tough measurement to take. It requires a lot of labor. It is like very, basically growers aren't really doing it. So we have low adoption. So we were wondering if a drone could maybe provide another alternative to that measurement. So that's kind of what we're doing here is I like this for a drone experiment because there are intentionally variable levels of stress. And that's what Erica Edwards here is a PhD student in our research group is monitoring. And we're also kind of thinking like, so there's the crop water stress index, and you would look at that over a block. And we've been kind of doing some work and thinking about maybe a crown water stress index for almond trees. Because there's literature out there that show that if you can isolate the crowns and the differences in the crowns with a tree crop or an orchard crop, that might give you a better estimate of stress than just looking at the entire thermal image. So how do you manipulate that thermal image to then make comparisons with crop water stress? So who asked about canopy temperature validation and calibration? Yes, okay, good question, and now we're there. So there are a couple different ways that you can do this. And also, once again, it all goes back to what is your question? Why are you doing this work, right? So if you're doing it because you want to make the best map that you can make, then it probably makes sense to try to develop some sort of calibration and then correct. Some of the work that we do, just like we talked about with the CIMIS, I'm interested in the variability and the uncertainty of not correcting. So sometimes we're actually intentionally not correcting because we want to see how well this tool performs without having to do that calibration. So that's how we're kind of making that decision. But you can actually put infrared thermometers out in the field and have some understanding of what pixel they correspond to. That's a very common way to do it. It's hard to see it. This is in a tomato crop in the 5 Points area of the Central Valley. You can see Logan has installed this infrared thermometer in the tomatoes that you can kind of see. Another way to do it, especially if you're using some sort of differential approach where you want to have variability in the scene and capture that variability, is by putting temperature or the same infrared thermometers, but over different surfaces, right, that are going to be large. And this is Utah State University Professor Alfonso Torres's setup for a project that we work on together, where we fly all of the drones and have all of the towers and all of the satellites at the same time in almond orchards. So some final thoughts. I changed this. I felt pretty cool because I used to say 10 years, but I feel like we're getting closer and closer to these in-season actionable evapotranspiration estimates and mapping with drones. And one of the biggest hiccups with this is actually the processing, right? We can go out and collect data quickly. It takes a long time to do the image processing. It takes a long time to do the mapping. And that's one of the things that's holding us back, but we keep getting better with that too. And then we talked a little bit about this. What types of different decision support tools are needed for different agroecosystems? It's not going to all be the same. You don't have to do the same amount of work for everything. It gets back to like, what is going to be actionable for the people who are using the data that you're collecting. And then I hope that we kind of brought this up inadvertently, just through sharing some of the work that we're doing. I think these collaborations, multi-institutional, public-private partnerships are really important to move this work along. And it seems like DroneCamp is a great example of that. Many people here are doing that. And with that, yeah, thank you so much. And any questions? I don't know when the next thing starts. Do we have time for a little bit? Yeah. Oh, yes, passing the mic. Oh, and I can check in. I don't know if there's Zoom people. I haven't looked to see if there's anything in the chat. Oh, cool. Yeah. Does anyone have any questions or things they would like to share or anything like that? Yes. Ooh, okay. What is a crown? It's like the crown of a tree. What is do you know the technical definition of a crown? I think of the crown as just that when you're looking over the tree, the top of the tree, like the top part of the tree. But that is not, if there is a tree person that has a much, much better definition than me, because I'm not a tree physiologist, of exactly what a crown is, but that's how we're defining it using the aerial imagery, is like, what is that concentrated part of the plant that we're able to image with our thermal imagery. But yeah, that's the best I can do. But I'm happy you asked me that, because now I want a more technical definition. I'm going to seek one out. Any other questions? Yes. Yeah, so ground control points, we just got a really neat tool that are called, what are those? Arrow points. We got these. Have anybody used arrow points or heard of them? They're pretty neat. They're about this big, maybe 1 meter. No, is it 1 meter by 1 meter? No, a half a meter by a half a meter. And they, you just turn them on and they connect to a variety of satellites. So I think they have RTK. It's like an RTK based model is what they said. PPK? Okay. They have PPK and they connect to a variety of satellites. They have like a positioning model that they use, that they're able to get a really good high resolution position. And then we have 10 of them, and then we'll set them up around all of the domain that we're trying to map. I want Logan to answer your other question, because I think he's experienced it more in terms of how timing the flight, this idea that the temperature is changing during the flight and all of that good stuff. Logan.
Yeah. So part of it is trying to fly within best practices. Typically for thermal imagery, you want to fly between 11 a.m. and 1 p.m. Back home in Wisconsin, it was a big issue with clouds, was a big cause of that. Trying to avoid, I guess the best thing is avoiding the situation where you're going to have those variability. At some point, you are going to have to accept some of it, unfortunately. Trying to keep your flights shorter is great. So going back to like flying really low if you don't need to is a thing that adds on to that. So if you have a long flight, you're going to suffer those kind of consequences of that changing temperature. Yeah, making sure, or not making sure, but if you want, you can try calibrating it with the methods that Mallika showed in those imagery. But yeah, just really flying at the right time, not flying when it's really windy or really cloudy is going to be your best bet.
Yeah.
And I
Oh, do you have a
I
mic?
can turn on my mic.
Okay.
Yes.
Sean's gonna opine.
So yes, I was going to echo exactly what Logan said about the time of the flight. I published a paper with some folks from UC Berkeley. We flew every half hour from first thing in the morning through the afternoon. We found that in the afternoon, when the stomata had closed up, the data was not really useful anymore. It was the morning hours that when it was the best. You also run into a little bit of saturation of the near infrared in particular, that was kind of the contrast between them became less important as the day went on into the afternoon. And again, flying high, because with thermal imagery, you got maybe a 20-minute flight. A good wind picks up halfway through your flight, and the thermal values are going to change on the ground really fast in a way that spectral values won't. So flying higher oftentimes will get you better data.
Yeah, absolutely. And the last comment on it is, if you are concerned about like the ramp up that occurred during your flight and you have, met data, you can use that to correct your flight, right? But that would be quite a bit of work when you think about the pixels and like your flight pattern and how you'd go about correcting. But I just want to bring up, it's possible to actually use the thermal changes that are occurring during a longer flight and make a correction. But good question. All right. Yeah. Oh, yes.
Kind of a policy question or big picture question. when you publish your results, or when you do the successful ET mapping results, are you able to use this actionable data for, let's say, the benefit of improved business decision making, public policy decision making? In the light of the fact in California, we do have now the Sustainable Groundwater Management Act, and SGMA is being planned as we speak. The groundwater basins are doing their plans. So I would imagine the California Farm Bureau Federation Foundation, as well as Kern County Farm Bureaus and supervisors would probably benefit if they knew exactly how to save water, increase yields, healthier crops. So how do you interact with those folks?
Yeah, what a great question. Okay, so just kind of to talk a little bit about that sustainability question, which is big, and how to integrate like our snapshot data into something like that. I brought up the project that Erica has worked on, and that Andy's worked on that as well. And we call it the T-Rex project, and it's the tree remote sensing of evapotranspiration experiment. And in that project, we're working with several different institutions, and we are integrating the drone data, the Flux Tower data, and the satellite data, and working with folks, for example, who have developed the OpenET tool, which is going to be something that is, very useful for policy in terms of being able to try to monitor evapotranspiration and water use all together, right? So the dream, I think, of a project like that, at least our dream on the T-Rex project, is to be able to have really high resolution continuous evapotranspiration data. And that could be used in this type of water policy work, right? Thinking about how much water is being used vastly over a landscape or in a water district. There's a lot of people asking questions about how much water cover crops are using. And this type of work can be used to kind of think about like how much water are we using in the off season or particular times, and those types of savings. Another kind of not sexy part of it is it's also useful to think about like when systems don't have good uniformity and the irrigation systems themselves aren't performing well, because then you start to see like predictable patterns that are based on the system providing a lot of water to one area and not to another area. So that's another way that they could be used to kind of inform better irrigation practices. But that's a great question. And I do think it's something that we are working on, and many people are working on, and very important to think about. And I'm going to plug my podcast because you brought this up. I have a podcast called Water Talk, and it's all about California water issues. And it's myself and Sam Sandoval and Faith Kearns. And we just think about like issues like SGMA at the state level. How do we integrate different pieces of scientific information, talking to a lot of experts? So I think I've gone over enough at this point.
It's okay.
Okay. All right. Thank you guys. Thank you so much.